very much to the organizers for this invitation to present my work, and thank you very much, Bjorn, for this introduction. It's quite nice for me to be able to, to at least virtually meet some local people interested in theoretical biology. So this is great. So I'm going to tell you about some work on microbial populations evolution. But before I begin this, let me tell you that my lab also works on protein sequence evolution. So, just in case you were wondering about general activities in the lab, we have mostly these two directions: evolution at the level of proteins and evolution at the level of microbial populations. And it's all theoretical. Okay. So my lab is called the Laboratory of Computational Biology and Theoretical Biophysics. It's affiliated with the Institute of Bioengineering and the School of Life Sciences at EPFL, also affiliated to the Swiss Institute of Bioinformatics. And today, the title of my talk is about the impact of population spatial structure on mutant fixation probabilities, first in models on graphs, which are quite abstract, and then in the concrete case of the gut. So I will start by telling you a bit about abstract models. So this is the first part of my talk, which is about toward the universal model for spatially structured populations. And this is joint work with my former PhD student, Loïc Marek, who just who defended his PhD recently and is now a postdoc in the University of Bern. And my former master student, Irene Lamberti, who is now a PhD student at EPFL. And so later on, I will tell you about the case of the gut. Okay, but first of all, let me introduce these models and graphs. And the motivation for all this is that spatial structure is very important for microbial populations. In fact, in the case of a lab experiment, you may want to consider this well-agitated beaker that is represented here. But this situation is not very realistic apart from the lab. Natural microbial populations are generally not well mixed, and they have complex structures. In particular, if you consider the case of an infection, you may have structure that is subdivided among the organs of the patient. If you consider an epidemic, you have propagation among the host, which also gives a complex structure. And even if you think about simpler lab settings, so I told you about this agitated beaker, but you may want to consider the Petri dish, and the Petri dish is already a case where the spatial structure is something that is quite complicated because it's a 2D system, and locally bacteria are competing more with their neighbors than with other bacteria that are far away. So motivated by this, some models have been introduced in order to describe populations with complex structures. And in particular, in this paper, Lieberman et al., from the group of Martin Nowak, there was a general model that was introduced, and it's a model that can represent, in principle, any population structure by a graph. And so the idea is that on each node of the graph, so here you have an example with 5 nodes, on each node of the graph, you have one individual, and on each edge of the graph, you have a probability of replacement. And it's probability that one individual that is born in node 1 replaces an individual in node 2, which is called W12 on this array here. Okay, and so, so far it seems rather general, but in fact, in order to make this more precise, we need to choose an update rule in the dynamics. And in particular, this update rule needs to preserve population size, because here we assume that there is exactly one individual per node of the graph at each time. And this means that, for instance, we may first decide to choose one individual that is dividing. And here it's illustrated in this plot. So the idea is that you choose the individual that divides proportionally to its fitness, which is called Fi here, the fitness of I. And then you are going to choose who the offspring of I is going to replace, with the rule that it can only replace its neighbors on the graph, so those to which it's connected in the graph. So here it's connected to J, for instance, and also to this other one, which is not named. So these are the two possibilities. And with the probability Wij, the offspring of I is going to replace J. Which means that if the two types that were in I and J were different initially, and here you had one in orange and one in green, so they were different, Then you have the composition of the population that changes after this one update. You now have two orange individuals and one green one, while initially you had only one orange one. Okay. So here, what is important is that we are doing selection on birth. So fitness is at play when you choose the individual that divides, while death is only chosen, by the fact that we have this weight of replacement. So it's selection on birth, and we have chosen first the individual that divides. Now we are going to look at another case, which is called the death-birth case. And in this case, you first choose the individual that dies instead of choosing the one that divides, and you choose it uniformly. And then fitness is going to come into play in order to choose who divides. So fitness is on birth, exactly as before, but you first choose the one that dies. So here we choose uniformly, and we may choose this one, J, that dies. Is with the probability 1 over n, where n is the total number of individuals, the total number of nodes on the graph. And then once you have chosen this individual to die, you need to know by who it's going to be replaced, sorry. And here we choose that it's going to be replaced by the offspring of I, which means that it's the same as above, except that it was chosen in the opposite order. And this choice of birth is now made proportionally to fitness Fi, but also to the replacement rate Wij. So for now, it looks extremely similar. Just two choices, which are just two assumptions on this microscopic dynamics, this update rule. We are going to see that it's important, in fact, to remember which one we are using, which is why I introduced them in detail here. But within these models, in this paper, it was asked what is the impact of graph structure on the probability that a mutant takes over the population. So let me tell you a bit more about this. So the idea is that we want to know, like, if there is one mutant that appears somewhere on the graph, we want to know what is the probability that everybody becomes mutant after a certain number, a large number of generations. So indeed, it's a finite population, which means that if you wait long enough, either you will remain with only mutants or with only wild type. This is assuming that there is no further mutation. We look at the outcome of one mutation. Okay, so in this paper, it was shown that there is a particular class of graphs in which there is no effect of the graph structure on fixation probability of a mutant. And this class of graph is called circulations. These are graphs, you can see three examples of circulations here. These are graphs that are such that in each vertex, the sum of the weights that are incoming to the vertex is the same as the sum of the weights that are outgoing from the vertex. So inflow and outflow are equal. And a particular case of the circulation is this click or complete graph where every node is connected to all others with the same weight. You also have this one, which is the circulation, where you have just, migrations in a circle. You can also have much more complex graphs, but which satisfy this property. And in all of them, the fixation probability of one mutant in a wild type population, which has n individual, where n is fixed, is going to be exactly the same. And it's going to be the same as in a well-mixed population. In fact, here, this complete graph is a well-mixed population, okay, where each individual is equivalent to the other ones, and they are all connected together with the same replacement probability. And the fixation probability in a well-mixed population is well known. It was, in fact, it was calculated within the framework of the Moran process here. And I'm giving you the formula because it's quite good to have it. So this is a fixation probability as a function of the ratio of the fitness of the wild type divided by the fitness of the mutants. And here you may remember that fitness is important to choose who is dividing. Okay. So you are dividing proportionally to this value f. If the mutant fitness is larger than the wild type fitness, here this ratio will be smaller than 1. Okay. So here you have this formula for the fixation probability, and this is a fixation probability such that you start with one mutant, which is anywhere in the graph, and you end up with only mutants. So one orange initially, and you end up with only orange guys at the end. Okay. So far things are a bit boring. We have this class of networks, and they are all the same. No change, no impact of structure on fixation probability. However, it turns out that other structures, which are, not circulations, and in particular, which have asymmetric migrations, because all cases with symmetric migrations are included in the circulation case. Some other structures can amplify or suppress natural selection. What does this mean? Well, it means that a beneficial mutant is going to be more likely to fix in these structures than in the well-mixed case, and a deleterious mutant is going to be less likely to fix in these structures than in the well-mixed case. And one of them, which is quite, which is, often, let's say, the example that is taken because it's one of the simplest structures that affects this fixation probability, is called the star. So the star is this particular graph where you have a center and some leaves, and the center is only linked to, the center is linked to all leaves, which means that it can replace any leaf. But each of the leaves is only linked to the center and not to the other leaves. So there is a strong asymmetry between the center and the leaves. All the leaves are equivalent, which means that all migration probabilities or replacement probabilities from center to leaves are the same for each leaf, and from leaf to center also. So we have only two parameters in the model, which are the replacement probability from the center to the leaf and from the leaf to the center. If we consider the birth-death dynamics, which means that we first choose an individual to divide and then it replaces another individual, we need to have this relationship on our replacement probabilities, which is that the sum over all points where the individual ends up has to be 1. This is just because when you start from I, you have to end up in another place, or in I. You have to end up somewhere. And so the probability of all this is 1. It's just a normalization condition. And because of this, we can find the actual values of the replacement probabilities between the center and the leaf, and the leaf and the center. And we find that W is equal to 1 over n minus 1, where n is the total number of individuals in the system from the center to the leaf, and W is 1 from the leaf to the center. So you see that there is quite a strong asymmetry, which is interesting. We will see later that actually it really matters. For now, it's just an observation. And when we include all this information, it's possible to calculate the fixation probability of a mutant in the star. And it turns out that the expression is quite complex, but that it can be simplified where the number of individuals becomes large. So n goes to infinity. And in this case, the formula, you may notice that it's quite reminiscent to the previous one, which I recall here, which was for the well-mixed population, except that now what plays the part of R before is R squared. And so, R is this ratio of fitnesses. It means that the differences of fitnesses are going to be exacerbated in the star compared to the well-mixed population. And in fact, when we look at the fixation probability versus this ratio, so here in fact it's versus 1 over R, which is the ratio Fm over Fw, so mutant over wild type. You can see indeed that if you compare the well-mixed case, which is the black dashed line, to the star in the birth-death process, which is the blue line, you can see that mutants that are fitter than the wild type, so those that are such that this ratio is larger than 1 here, they fixed more likely than in the well-mixed case, while the mutants that are deleterious fix less likely in the star than in the well-mixed case. So this is what is called an amplifier of natural selection. Okay, so far it seems like the star may be an amplifier of natural selection, but in fact, it turns out that it's a bit more complicated than that, because if we study the opposite dynamics, which is the death-birth dynamics, then we find the opposite result. So this is what you can see on this plot. So here you have the same star with 10 individuals, which was studied in simulations. And we can in fact do some analytics, but this is simulation results. And you can see that while the blue curve is amplifying selection with respect to the well-mixed case, the red curve is rather suppressing selection. What I mean is that the fixation probability in the death-birth case is going to be lower for a beneficial mutant than in the well-mixed case, and it's going to be higher for a deleterious mutant than in the well-mixed case. So it's quite interesting to see that this microscopic choice of the model, which is really a modeling choice, changes really the outcome in a dramatic way, that the star can be an amplifier or a suppressor of selection, depending on this choice. It's all the more interesting that if you consider a natural population, a real microbial population, well, in general, it doesn't have to follow such a strict update rule. Like microbes don't really need to ask their neighbors if they are dividing in order to die or reciprocally, right? So it means that it would be better to have something that is independent of these update rules. Another point that we may also consider is that even though microbial populations very often have complex structures, uh, it's a bit hierarchical in the sense that locally they may be considered as competing freely, and then at a larger scale, they may be considered as not competing as freely. So we may want to have a coarse-grained version of this model where at each node you have rather a subpopulation, a DEAM, a well-mixed population, instead of just one individual. In fact, I should say that this has quite a long history, and here I have not cited all the papers, but in fact, these models with subpopulations have been studied before, not with complex graphs, so in a different context, rather with simple graphs. For instance, the fully connected graph has been studied. It's called the island model. But here we want to make this coarse-grained version for any complex graph. And I will also mention that there is actually a paper that did this coarse graining on complex graphs, which is a very interesting one. And what we are going to do is a bit different because we are also going to lift the constraint that population size has to be fixed, where in this paper it was done at a fixed population size. And our motivation for lifting the fixed population size is that we don't want to have events that couple birth, death, and migration between the DEAMs. We want all these events to be independent. Okay, so now here is a slide that presents the model that we built. And we started with something that is similar to the model on graphs. So we put D well-mixed DEAMs. And so instead of individuals, now it's DEAMs. A DEAM is a subpopulation, which is well-mixed, okay? And it's indexed by I, which is between 1 and D. Each of the DEAM has a carrying capacity, which is its maximal size, which is called Ki. And so Ki can depend on I. However, in what follows, I will mainly focus on the case where all the Ki's are the same, but we can generalize, which is why I written it here. And the number of individuals Ni in each DEAM can vary. We also have migration rates per individual along the edges of the graph, which play the part in a way of a Wij in the previous model, except that now it's migration, which can be separated from birth and death. And so now I'm coming exactly to these events, which are birth, death, and migration, and that can be independent. So we assume that birth is following a logistic growth rate. So the growth rate is the fitness times 1 minus Ni over Ki. And so this is the logistic regulation term. Here, my alpha is denoting the fact that the fitness can be different between the wild type and the mutants. And we have a death rate that is constant and that is the same for everybody. So selection is at birth. We could generalize, but we are considering this for now. This means that at steady state, if you consider at least the deterministic limits, you would have a size of the DEAMs, which is obtained by equating the death rate and the birth rate, and it gives K times 1 minus G over F if you have only one type. And in addition to birth and death, we have migration events, which are independent from them. And this is very different from the previous models. And then we are choosing to work in the regime such that DEAM sizes are close to steady states. So we are removing the constant size hypothesis, but not too much. We are just allowing DEAM sizes to fluctuate, but we are not allowing extinctions, etc., which would, make some very interesting effects, but that's a bit beyond what we want to study. So here we are working at almost constant size. We are also going to assume that migrations are rare. And the reason why we do this is that we want the fixation of one type, either the mutant or the wild type in a given DEAM, to be much faster than the migration timescales. The motivation for it is quite, to simplify things, just because here we are going to be able to describe the state of the population simply just by the number of DIMs that are mutants or wild type. And in this framework, mathematically, we can describe the system as a Markov chain, where events are migrations that can be followed by fixation. And in this regime of rare migrations, it's simpler to describe the system. Okay. Then we are going to ask whether one mutant is going to fix or not. And we are going to start from one mutant in a given DIM, which means that it's going to first fix in its DIM and then spread to the rest, because we are in the rare migration limits. And because the fixation in a well-mixed DIM is already well known, I mean, this is a formula I showed you in my introduction before, we are going to focus on the spreading step. So we are going to start, in fact, from an initial condition where one DIM is fully mutant. And we are going to choose this DIM proportionally to its size, which is something that is realistic if mutations happen uniformly at random or upon division. In both of these cases, this would be the realistic thing. Okay. So now let me show you some structures that we are going to talk about. So the first one is the clique or the well-mixed graph. I've already introduced it in the previous model. It's the case where all DIMs are equivalent, all DIMs are connected with the same migration rate. So it's the simplest possible structure. The next one is the cycle, where you can have different migration rates in the clockwise and anti-clockwise direction. And we are going to consider the star, motivated by the previous studies that show that it can be an amplifier or a suppressor of selection, depending on the dynamics. I will not go into the details of the doublet, but this is just to show you that, in fact, our model can be extended to different population sizes, different DIM sizes. Okay, so we also start from, as I said, from one mutant DIM. It's here in blue, while wild type is in orange. So this is our initial condition. And now I'm going to show you the results that we obtained. And this is for the case of the clique, so our fully symmetric and connected graph. Here, I'm showing you the fixation probability of a mutant versus the fitness of a mutant. And this is in the case where the wild type fitness is 1. Sorry, I heard a noise. Maybe there is a question? No? Okay, I mean, maybe it was.
I didn't notice.
Okay, fine. So maybe it was just a random noise. No worries. I mean, but if there are questions, I'm very happy to take them. Even during the talk is fine. So feel free to interrupt. Okay. So with this, let me go back to this plot. So as I said, the wild type fitness here is 1, which means that mutant fitness, if it's larger than 1, is beneficial. If it's smaller than 1, is deleterious. And you can see here the fixation probability versus mutant fitness. We are plotting the case of the clique, which is our structure, and the well-mixed case, which is a population without any structure. You can see that it seems very similar. Okay. So, here I should say also that we have simulations of the process, which are done with the Gillespie algorithm, and these are the markers. And we also have a theoretical calculation, which is a fully analytical expression. And basically what this is telling us is, however we look at it, we have essentially the same. So let me explain a little bit how we did this. We use the fact that it's a Markov chain in order to express the fixation probability. And we looked at the probability that the number of mutant DIMs, which is named I here, is going to increase upon a migration event. So I won't go into the details, but this is like the probability that I increases by 1 when you have a migration event. And then by using these probabilities that the number of mutant DIMs increases or decreases upon a migration event, you can find a recurrence relationship on the mutant fixation probability that can be solved. And it means that we have this analytical formula for the fixation probability of a mutant in the clique. Here, what is quite interesting about this formula is that it's formally quite similar to the one that you may remember from the well-mixed case. The only differences are that here the number of DIMs is playing the part of the number of individuals, and that instead of the ratio of fitnesses, we now have the ratio of fixation probabilities the rows are the fixation probabilities times the equilibrium sizes when you have a mutant and when you have a wild type. So it seems to be formally similar. I will come back on this formal analogy later on. But now let me show you the case of the cycle. And here you can see the case of the cycle for various values of the migration asymmetry, which is the ratio between the anti-clockwise and the clockwise migration rates. You can see here our simulation results in different colors, but what you see is that essentially they are all doing the same. It doesn't seem to depend on the value of mu. And in fact, if we do the same kind of recurrence relation as before, we can find the theoretical prediction, which is in gray here, and which does not depend indeed on mu. Okay, so we in fact find the exact same formula as for the click in this case, with the same expression of gamma, and this gamma has the ratio of the fixation probabilities and of the steady state sizes. Okay. So that's, for now, everything seems to be very like boring, let's say. All these structures do not impact fixation probability, essentially. They are very close to the well-mixed case. I'm saying very close because, in fact, if you look at the details, there is a very minor difference, but it's really very minor. Okay. And the click and the cycle have exactly the same one. All right. But so now we decided to next look at the star. And in the case of the star, as you can see on this plot, we are again plotting fixation probability of the mutant versus its fitness. As you can see now, we have different colors of curves, which have different shapes. And this corresponds to different asymmetries of migrations, which here are the ratios of incoming migration to the center to outgoing migration from the center. So it's what is coming in versus coming out. And we can see that this impacts a lot the behavior of the fixation probability in the star. And in fact, again, it's possible to find in this regime of rare migrations, it's possible to find an analytical expression for the fixation probability in the star. And this time, we have indeed a dependence on mu, which is our migration rate asymmetry. And it's possible also to look at the limit where mu is large, so the limit of very asymmetric migrations such that, so mi is much larger than mo. And in this case, quite interestingly, we find something that is formally very similar to the fixation probability in the case of the well-mixed, in the case of the star, in the case of the star for the model where you have only one individual per node. Okay, so it's very similar, except that, again, we have our ratio gamma that replaces the ratio of fitnesses. And this is for the large D, in the case of the large D limits, you would really have something that is more similar even to this approximation, which itself was obtained for the large N limit, so it makes sense. Okay, so that's the star. Because we see that the formula seems to be simple for large D, and because we see that there may be more amplification of selection for large D, because there was more amplification for large N in this case, we also looked at the case of a star which has more dims. So here we had a small star with only 4 leaves. This one has 100 dims, so 99 leaves on one center. And you can see here that the results are even more striking. If mu is larger than 1, you have something where essentially selection is amplified. So beneficial mutants fix more likely than in the well-mixed case, while if you have mu smaller than 1, they fix less likely than in the well-mixed case. So it means that the star transitions from being an amplifier of selection for large mu to being a suppressor of selection for small mu. So here it really seems that migration asymmetry is a very important parameter of our model. And for mu equals to 1, we have the case where it's like structure does not matter. We are back to the circulation theorem, essentially. Okay, so these are our results for the star. And we may now wonder why migration asymmetry is so important. So let's think about this a little bit. And mu here is the ratio of the incoming to outgoing migrations from the center. And if mu is large, it means that migrations from leaves to the center are more frequent than the opposites. while if mu is small, it means the contrary. If the number of dims is large, because our mutants are placed randomly initially, it means that most of the mutants arise in leaves and not in the center. And the large mu means that these mutants that start in a leaf, so for instance here, they can spread via the center because, the incoming migration rate is big, but then they are in competition with other migrants that may come from other leaves, right? While if you have a small mu, it means that it's difficult for these mutants which are in the leaf to spread in any case. So there is one exception to all this, which is the case where a mutant arises in the center, which again is quite rare. If a mutant arises in the center, it spreads well for small mu, and in fact there is almost no competition for it, which means that in this case, this mutant from the center is why if mu goes to 0, we have this asymptotic formula. Okay, so the idea behind this importance of asymmetry is really this, like in one case, you favor the mutants coming from the leaves, and in the other case, those coming from the center, but there are more that come from the leaves. Okay, so yeah, I was saying that in the star we have this. Okay, so I will go fast over this path. We have been able to show that there is actually a mapping to the previous model. So the model that was introduced in the paper by Lieberman and colleagues here was such that you had only one individual per node, and our model has a dim in each node. It turns out that in the framework of this rare migration regime, we can make a quite complete analogy by looking at the probability that the progeny of I replaces J in this model with one individual per node, and the probability that an individual from I fixes in J in our model. And it turns out that it's formally really very similar. And it means that, in fact, we find a general formal mapping where the fitness in this model is replaced by the product of population size and fixation probability in our model. And the replacement probability is replaced by migration rate. This means that, in fact, you may remember that I told you for the case of the click on the cycle, we have the same form of fixation probability, but with gamma being this ratio that replaces the ratio of fitnesses. So this is completely explained by this general mapping. It also means that we have a generalized circulation theorem that holds for our model, which means that in the case of a circulation, you will find the same probability of fixation as for the click. And in fact, you may remember the case about the cycle. So the cycle here had exactly the same fixation probability as the click. It's because the cycle is a circulation. Okay, so we have this analogy, but nevertheless, we have a difference, a major difference with the previous model, which is that here we don't have any constraints of fixed size that is imposed. And it means that we have no requirement that is equivalent to this sum over the replacement probabilities that has to be equal to 1 in the birth-death case. And in the case of the star, if we look at what it means in fact that if we impose this, we have d minus 1, so the number of dims minus 1, that has to replace the migration asymmetry mu. In this case, we actually recover the formula of the model with one individual per dim. And so with this, I would like to show you a heat map that shows the ratio of fixation probability in the star divided by that of the well-mixed the well-mixed population, and it's plotted versus migration asymmetry mu and number of dims d. And what you can see here is that there is a whole range of parameters where this ratio is smaller than 1, so in the red region, and another range where it's blue and larger than 1. So the star really transitions from being an amplifier to a suppressor depending on mu. And you can see also here this white line, which is the case where mu is equal to d minus 1. And this is the case where we recover exactly the results of the birth-death model in the case with one individual per d. And this explains why in the birth-death model, in fact, you always see amplification and you never see suppression. This is because you have constrained this migration asymmetry by using the update rule. And this migration asymmetry is crucial to determining whether you have suppression or amplification within the given structure of the star. Okay, so with this, I will summarize this first part. So we found that we developed a coarse-grained model that lifts the fixed size constraints, and in our model, migration, birth, and death are independent. And thanks to this, it elucidates the apparent paradox in constant size models where amplification or suppression depends on the update rule. Here, what matters is the migration rate asymmetry. So this study could be extended to other graphs, different dim sizes, which is something that we already did, but I don't really have time to talk about it. We can also go beyond rare migration, and I think this would be interesting, and we can study fixation times in addition to fixation probabilities, which would be very relevant to the rate of evolution. It would be interesting also to make a more concrete link to experiments, specifically in the case of microfluidics or microtidal plates. In fact, there are some people who are working on such systems. The impact of population subdivision in general is quite interesting, and it could impact, for instance, the evolution of antibiotic resistance. So with this, I'd like to thank my co-workers on this project, who are two students, so Loïc Marek and Irene Lamberti. And if you want to know more, we actually have a preprint about this. And this project was partly funded by the ERC. So I see that time is flying very fast, but I'm happy to take questions on this first part, if there are any extra questions. Okay, so if not, I will do a brief second part. So I will try to go fast on this, and please let me know if it's getting too long. So my second part is about the fate of neutral bacterial mutants in the gut. And this is joint work with my collaborator, Claude Lauverdeau, from Sorbonne University in Paris, and our postdoc, Darka Labavic. Okay, so the motivation of this part is that the gut microbiota is very important, and in fact, there are as many bacteria as human cells in the human body. 99% of these bacteria being in the gut, mainly in the colon, and there is a high diversity of species in this microbiota. One source of this diversity being possible evolution in the gut. Of course, you can have incoming diversity, but here we are going to focus on evolution in the gut. The microbiota has important public health implications. In particular, the gut can be a reservoir of drug resistance in humans and animals, so evolution in the gut could be important for this. And here we are going to consider the gut as an environment for bacterial evolution. We are going to take into account the fact that there is a flow in the gut, which is along the main axis of the gut. And we are going to focus on those bacteria that are carried with the flow of the gut. In fact, there are some bacteria that are associated to the mucus or that are in crypts. We will not focus on those. We will just look at those that are carried with the flow. And these are, in fact, the most abundant bacteria in the gut. The flow of the gut is quite complex. There are, in fact, contractions of muscles. That means that the digesta is mixed by these contractions. And it also means that it's hard to model. There are also gradients of food and bacterial concentration. Typically, in the small intestine, the bacterial concentration is very low, which means that at the entrance of the large intestine of the colon, you have a small bacterial concentration, but then the concentration grows along the axis while food is being consumed. So you have these strong gradients and the contractions. So now it seems that it's a system that is very hard to model, but it turns out that it was proved in this paper that actually has modeling and also an experiment of a mini gut that is quite reproducing some main features of the gut. It was proved that the contractions can be modeled in a coarse-grained way as an effective diffusion, which is much larger than the real diffusion coefficient of the system. It's just something that models mixing. So we are going to take this into account, and we have the simple ingredients that there is a flow and a diffusion. We also consider that at the entrance, we have food that comes in, and we focus on the entrance of the colon, and then we have a free outflow to the rest of the colon. So we focus on the segment at the entrance of the colon, and we are going to ask what is the impact of flow and mixing, effective diffusion, on the evolution of gut bacteria. Okay, so I will go fast on the model. I will just tell you that we considered a model again based on this paper, which is based on partial differential equations that describe the concentration of food, F, of bacteria, B, and you have diffusion and flow, which are the ingredients here. And also here you have this term, that is the reproduction term. So bacteria is reproducing by using food. So that's the interaction between food and bacteria, the consumption of resources, it's in this term. This term has a maximum reproduction rate, a saturation, such that you have a maximum possible growth rate, R. And this saturation is implemented by this denominator here with the monoconstant K. And there is also a factor alpha, which gives the conversion from food to bacteria. So we have these coupled partial differential equations. We also take into account boundary conditions such that you have an inflow of food, VFN. You have no inflow of bacteria, 0. And at the exit, x equals L, we have free outflow. So the diffusive flux is 0. Okay, so this is the kind of model we are looking at. And we have done a numerical resolution of this system. And here you can see the profile of the food concentration versus position along the gut. What happens is that initially you have a lot of food, and then food gets eaten, and you have like low food concentration. Accordingly, bacteria is doing the symmetric thing, it's growing. And we can also look at the reproduction rate of bacteria, which is R times F divided by K plus F. It's in red here. You can see that it's also very strongly spatial. You have strong reproduction close to the entrance and then low reproduction. So this plot was made for a specific value of velocity and diffusion. And we may wonder how generic this is when these values are varied. For this, we quantified spatial dependence by the ratio food at the entrance minus food at the exit divided by the incoming food concentration Fin. And you can see here a heat map of this quantity, which quantifies the spatialness of the profiles versus diffusion and velocity. What you can see is that there is a big zone of parameters where the concentration profiles are very flat. This is the purple zone. And there is only this region, this particular region, where you have strong spatial dependence. So in fact, concretely, what happens if you are in the purple zone to the right is that all bacteria are pushed out of the system by the flow. It's called the washout regime. Here you have something that is almost well mixed because velocities are small. And here you have this intermediate case where things are spatial. And this is in fact something that is closer to what we would expect in the real gut. Because in the real gut, there are low bacterial concentrations at the entrance of the colon, and then it gets larger. So we will focus on this regime, and it turns out that it can be checked that the parameters are consistent with those of the actual gut. Okay, and so what we did was to introduce neutral mutants in this system. For this, we add one extra equation on the mutant, which is exactly the same as for the wild type. And this means that at steady state, we in fact have a ratio of mutant to wild type, which is constant, not dependent on the position X. And the question we ask is, what is this ratio going to be, and how is it going to depend on the initial conditions of the mutant? In particular, we are going to ask how it depends on where the mutant starts. The mutant can start close to the entrance, it can start close to the exit, and this may matter for this ratio M over B. And here you can see a plot of M over B versus the mutant introduction position XM. And what you can see is that the mutants that give the largest ratio M over B are those that appear close to the entrance. And this makes sense because they have more opportunity to spread and divide in the system. And in fact, here, this is a deterministic description. We have done like partial differential equations, etc. But M over B will also give the fixation priority of these neutral mutants if we were to look at a stochastic system. So, M over B is really an important quantity, but it's not the whole story because, mutants that appear close to the entrance are more likely to fix, but they may not be the most likely to appear. So what matters is where they appear and then if they fix. And so we ask the question where mutants that fix tend to originate, which depends also on reproduction, if we assume that mutants appear upon reproduction. And so here I'm plotting not only M over B, which is the black curve, but also the reproductions per unit volume and per unit time, which is called R here. And you can see that R has a maximum at some intermediate points. This is because in order to have a big value of this number of reproductions per unit volume and per unit time, you need to have a big enough bacterial concentration and a big enough reproduction rate, which depends on food. So close to the entrance, bacteria concentration is small, which means that R is small, and close to the exit, food concentration is small, which means that R is small too. So the ideal is an intermediate position, and this means that these intermediate positions are going to be important. And if we want to find out where in the gut the mutants that tend to fix will originate, we need in fact to look at the products of their appearance, which is given by R, and their fixation priority, which is given by M over B. And so we look at these products of R and M over B, and it turns out that it also has a maximum at an intermediate position, which means that these are the ones that will matter most. And so, okay, I will probably just show you this figure and then I will stop. So what we looked at next was the overall fixation priority of a mutant that may appear upon division. So it may appear anywhere in the system, proportionally to the number of divisions that there are at this point, which is why we include R here. And then its fixation priority is M over B. And so we have to, okay, this is just normalization. And so this is a fixation priority, and we want to compare it to the well-mixed case. And the well-mixed case would be just a fixation priority that is proportional to the number of mutants divided by the total number of individuals in the system. Here, we want to ask how F is going to be impacted by spatial dependencies in the gut. And previously, we introduced a measure of spatial dependence, which was this ratio of like food at the entrance minus food at the exit divided by incoming food. So we are going to see how F depends on this. And here are the results. So here on the right-hand side, this is just a reminder of how diffusion and velocity impact this spatialness of the system. You can see that, okay, there is a region where the system is very spatial, and in the rest, it's not very spatial. And in fact, these colors are used on my left-hand side plot. So on the left-hand side, you can see fixation priority versus total population size of the system. And the light blue line represents the well-mixed expectation, which is this, NM over NT. And the dots correspond to the results we find in our numerical resolution of the system. And you can see that there is a strong deviation of the fixation priority compared to the well-mixed expectation, especially in the cases that are strongly spatial. You see here, these red points are those that are the most spatial. And for those, fixation priority becomes independent. It's a plateau. It becomes independent of population size. So it means that the structure in the gut with the flow and the diffusion that represent mixing and the resulting gradients of food and bacterial concentration can have a strong impact on the outcome of a neutral mutant. We also showed, in fact, that this can be understood by using the fact that only a part of the population is actively reproducing. So only the population that is sufficiently close to the entrance has a large reproduction rate, because afterwards food is depleted. And we actually showed that this can quantitatively explain our fixation priority. I won't go into the details because I think I'm lacking time. But yeah, this kind of explains this interesting result that fixation priority is no longer proportional to total population. And so just to conclude on this, we found that the gut features strong spatial dependencies of food and bacterial concentration in agreement with the model that was developed in the paper by Kramer and collaborators. And the fixation priority of a neutral mutant, we find that it can be substantially larger than in a well-mixed system with the same total number of bacteria. And this means that the spatial structure of the gut may favor the spread of neutral mutants and the evolution of bacterial population composition. So it would be interesting to extend this to the case of beneficial and deleterious mutants. So far, it's just neutral mutants, okay? But what is quite interesting is that in the first part of my talk, I told you that you have amplifiers or suppressors of selection, etc., but none of them ever changed the fixation priority of a neutral mutant. This one was always staying constant. And here, with this complex structure, with the gradients, etc., we have a different priority of fixation for neutral mutants. So that is, I think, an interesting point that relates to two parts. It would also be interesting to make a more detailed model of transport in the gut, going beyond this effective diffusion that models the contraction. I think there are lots of things that could be done here. And also to model potentially the interactions between the bacteria in the mucus and those in the lumen. Here we just focus on those in the lumen, which are the main ones that are flowing along the content of the gut. So with this, I'd like to thank my collaborators on this project. First of all, Darka Labavich, who was our joint postdoc between Claude and I, and second, Claude Loverdo, my collaborator from Laboratoire Jean Perrin of CNRS and Sorbonne Université, which was actually my former institution. And we will have a preprint about this very soon, but it's not exactly there yet. And we got funding from Sorbonne for this project. And finally, I would like to thank, so I already thanked these collaborators on the project, so Claude, Darka, Loïc, and Irene. And I would like also to thank other colleagues for great discussions, in particular David Schwab and Raphaël Guatroyer. And I also take the opportunity to thank my current group members, even though in this talk I did not present their work, but some of it is related, and my funding sources, which are currently EPFL and a starting round from the ERC. And with this, I would like to thank you very much for your attention. And I'm happy to take any extra questions that you may have, even though there were already questions during the talk.